Hope everyone's having a good Friday. Hey, how are you, Dr. Rice?
I'm good. I go by Charlie. Are you?
Okay, we'll go with Charlie. How was the logging on? I know I was talking with Santa early in the week. Was the logging on pretty smooth? Can you hear me?
Well, since Santa is, make sure that it works okay. It was smooth.
All right, wonderful. Well, listen, thanks for joining us. I know you're super busy. I'm going to do a brief introduction just while everyone's logging on here. So we are lucky to have Dr. Charlie Rice. He is one of the world's leading scientists. He won the 2020 Nobel Prize for Medicine or Physiology for his work on discovering the hepatitis C virus and all of its treatments. In brief, his work has generated a version of hepatitis C that could be studied in the laboratory for the first time, enabling the development of antiviral drugs that were capable of reducing hepatitis C down to undetectable levels, essentially making it into a chronic disease as opposed to a terminal disease. As background, Dr. Rice, or Charlie as he likes to be called, attended UC Davis for college and then Caltech for graduate school, where he focused his research on RNA viruses. At Caltech, he deduced the genome of the virus that causes yellow fever, and he helped develop a subsequent yellow virus vaccine. He also helped establish the flavivirus family, which includes the well-known West Nile virus, as well as dengue fever. Dr. Rice has then also joined the faculty at Washington University after leaving Caltech, where he shifted his focus to the development of a vaccine for hepatitis C, and he has since worked on that. He generated a culturable version of the virus, allowing him to provide a description of the genome and then demonstrate the infectious nature of the virus. In 2001, Charlie moved to Rockefeller University, where he made several other key findings, amongst them the discovery of how hepatitis C actually does infect liver cells, what is the biomechanics of that, and the design of multiple new therapeutic agents for hepatitis C. The first of these drugs was approved in 2013 by the FDA. Dr. Rice is the recipient of the 2015 Robert Koch Prize and the 2016 Lasker DeBakey Clinical Medical Research Award. He was an elected member of the American Association for the Advancement of Science, as well as the National Academy of Sciences. So truly one of the world's most brilliant scientific minds. Charlie, thanks for joining us. It's really an honor to have you on with us today.
It's a pleasure to be here.
Wonderful. And I know you're at Rockefeller, which is right across the street from where I did my fellowship. I was up at Sloan Kettering, so hope everything's going well in New York.
Well, it's starting to return to a little bit more of a normal-like state. I wouldn't say we're completely there yet, but no, the city is livening up. So you would probably recognize, some of the old signs of New York City. It was certainly a different place, sort of being the epicenter of the pandemic here in the U.S.
Yep, and we'll get into that in just a little bit because I want to hear about how your lab helped out with COVID. But tell us just how did you find out that you won the Nobel Prize? I always hear these stories, and they're always kind of comical because they catch you off guard. But how did you find out?
Well, I'm kind of more of a late night person, and so when the phone in our apartment sort of rang at 4:30 in the morning on October 5th last year, I was basically just pissed off that I thought for sure it was a crank phone call, and I ignored it initially and sort of head back into the bedroom after it stopped ringing, and then it rang again, and I sort of stumbled out into the living room and grabbed this landline phone, and there was the secretary of the Nobel Prize Committee, sort of a gentleman with a Swedish accent on the other end of the phone. But I really, I was not expecting this, and so I was in a state of disbelief, and maybe I still am. But when he began to talk about hepatitis C and Harvey Alter and Michael Houghton, who were the sort of co-laureates for the prize in 2020, it started to make a little bit more sense. But it came as a complete surprise.
I mean, what's the first thing you did when it finally set in that you had won the Nobel Prize?
Well, I actually, I sort of did a few things before I think it actually set in. You know, obviously, if you get a call like that at 4:30 in the morning, you're not going to crawl back into bed and, you know, sort of go. You're probably already up at, you know, sort of 4:30 in the morning. You know,
Kind
sort
of,
the
absolutely
neuros
prime time.
but I wasn't. So I sort of decided to take a shower, make a pot of coffee, called my spouse, who was actually not in the city, but was sheltering with our dogs at a house in Connecticut. And then I waited for the formal announcement, you know, which came about an hour after the call did. So I sort of logged into the Nobel Prize website and sort of watched that. And then it seemed at that point that it was pretty much, it was true.
Wow. I mean, so for the 99.9999% of the world that will never feel that sense of accomplishment that you went through, can you try to describe what it feels like to work your entire career, you know, obviously very well accomplished, and then to hit something like that is such a legendary, epic achievement, the sense of fulfillment, the sense of accomplishment, the sense of teamwork?
Well, I would say for me, it was really the sort of the last part of, you know, sort of what you said, the teamwork. You know, I really didn't and don't view this as sort of a sign of personal accomplishment per se, because as you know, any advance in biomedicine involves, you know, sort of a lot of history, a lot of people, so at least for me, it wasn't like, oh, wow, you know, I won the Nobel Prize. It was more like, you know, hepatitis C, a lot of people worked on this for decades to sort of get where we are today. It was nice to have, you know, those accomplishments recognized with the prize. And I think for me, the most important thing about it really is to, you know, sort of you're making this, you know, sort of more public, you know, by sort of doing this interview, but it provides an example of what a concerted effort in medical research can bring in terms of a significant advance for people. So that was, you know, for me, who is a very much a fundamental, you know, sort of basic scientist, to have sort of gone through that journey over several decades and having had it, you know, sort of end up where we are today in terms of having these miraculous drugs, you know, that really is the prize, if you will.
Yeah. And you talked about team. How did you tell your team, and what does the Nobel Prize mean to your team?
Well, they were pretty excited, you know, but this did come at an odd time when we were basically, the university was pretty much, you know, still in a partial shutdown state, you know, and we were distancing and wearing masks. And so the same kind of sort of team celebration that you might envision for this was quite different. We actually did have one that involved not only current members of the lab, but people that have left the lab, alumni, that we had via Zoom, which was actually a lot of fun because it brought a lot of people out of the woodwork that I hadn't seen in a long time, because this has been a very long journey. So yeah, there were some more limited, I would say, celebrations than there would have been under other circumstances. It was an interesting convergence of viruses, I say.
Yeah, absolutely. And you know, winning the Nobel Prize comes with a financial prize as well, but you've been quoted as saying you're not going to use it for anything personal. Have you decided what you're going to use the Nobel Prize finances for?
You know, I've been so busy that I haven't actually decided yet, but you know, I'd like to do something that would be, you know, sort of a lasting, sort of positive effect for biomedical science. So, you know, maybe a lectureship, or, you know, a special fund to allow sort of an expansion of diversity, either at Rockefeller or at one of the other institutions that I've been associated with. Still thinking about it. So if you have lots of good ideas, I mean, people would say, okay, just write me a check. But if you have good ideas, I'd be happy to hear them.
Yeah, I mean, it seems like with that money, you could really form a legacy. You could do something that just continues to give back to one of these prestigious universities you've been at, just for, just for the long haul, and kind of have that legacy based off your achievements. But, let's go back to your journey, because I find it so fascinating. Why did you pick basic science, and why virology? Obviously, you're brilliant. You could have done anything you wanted in terms of science, but why virology?
Well, it was a series of random collisions. I don't know if you always wanted to be a doctor and a surgeon or whatever, but I think when I, finished high school, I really wasn't, sure what I wanted to do. So I went off to UC Davis, maybe with the thought of becoming a veterinarian. That UC Davis has a good vet school, but it was really introductory biology class taught by Dennis Barrett. It really got me excited about biology. And he actually became a very important sort of mentor to me. And one of the things that, there were a couple of things that he catalyzed. One of them was spending a summer at Woods Hole at the Marine Biology Laboratory, where if you take one of the courses there, you're just completely immersed in science and research, basically 24/7, for the summer. And for me, that was, sort of a transformative experience, really cementing that I liked, science and research. And then in terms of how virology, so at Woods Hole and also at UC Davis, the research that I did was actually on sea urchin development. Because you can fertilize sea urchin eggs, and you can just sort of watch them develop in seawater. It's just an amazing thing to watch. And so I was really interested in developmental biology, and that was one of the reasons why I applied to Caltech. But when I got to Caltech, I didn't end up being placed in a developmental biology lab. I ended up in Jim and Ellen Strauss's lab that studied animal viruses. And so it was really just, kind of this random placement that, sort of set me on this sort of virology path, which I've never deviated from since then.
Wow. Who would you say is I know you mentioned one of your professors, but over the course of your entire career, who would you say has been your biggest mentor and role model?
Ooh, I don't know. I mean, I do think that, sort of Dennis, sort of my introductory biology teacher, and also the person that I did undergraduate research with was really sort of an important sort of mentor and role model. And my advisors at Caltech as well. I mean, there's so many other, sort of scientists and physician scientists that, one is influenced by. It's hard for me to, just sort of point the finger at any given person. There were many people at Caltech, investigators there that, just had a real passion for sort of discovery. And I think that kind of atmosphere and being surrounded by those kinds of people just helps to sort of instill the same, sort of goals in someone. But,
yeah, I mean, I think no matter what we do, whether it's basic science, it's clinical medicine, law, business, we all have role models. We all have mentors, people who kind of inspire us. How important is that for you to do that for your students? I mean, obviously, at your stage in the game, you have multiple students, postdocs, people who you're mentoring. How important is that for your legacy to you?
Oh, I think that is the most important legacy, because I think anybody's individual accomplishments, are what they are. And when you're gone, they're static. But I mean, if you really sort of instilled some of that enthusiasm and know-how and desire in people that have trained with you, that's really, those are like your children, at least for me. And so it's very important. And that's what I spend a lot of my time, sort of thinking about and worrying about is, making sure that, people have a productive stint in the lab, and also that they're positioned in the right place to go where they want to go after they leave here. So, yeah, I view that as being sort of the, my main duty right now, my main focus.
Yeah, I think we all at some point in our career need to realize that having that legacy following you is more important than what you've done yourself, because that's obviously going to end at some point. But having people follow you who understand your values and kind of what your goals are is critical. I want to get into a little bit about the basic science about your hepatitis C variant, which you helped, form a version which was able to be replicated in the lab, which had not been done before. What was the key discovery that allowed you to culture this virus where it couldn't be cultured before?
Well, there were a lot of things. I mean, we knew from work that, Harvey Alter and others had done that this agent of post-transfusion non-A, non-B hepatitis, which became called hepatitis C in 1989, could be used to experimentally infect chimpanzees. The problem was getting it to actually sort of replicate in the laboratory, and also just sort of coming up with a toolbox for studying this virus that was like other positive strand RNA viruses. And this is maybe a little bit difficult to explain in sort of a short few sentences, but one of the things that we do with these RNA viruses, and this includes SARS-CoV-2, is to actually sort of force them through an artificial DNA intermediate. So we make a complementary DNA copy of the viral genome, and then we can amplify that in the laboratory, and then you can use that to make RNA molecules that actually are synthetic mimics of the actual genome RNA of the virus. And so this was one of the goals that many of us had in the sort of early days of hepatitis C research. But we had a problem there, and that is that if you want to test the ability of these RNAs to actually initiate the complete replication cycle of the virus, you have to be able to get them into a cell that the virus can infect and replicate and make progeny in. And we didn't have that for hepatitis C. That was frustrating. So we were left with the way of testing these. It was, and you'll appreciate this, was actually just intrahepatic injection, injecting this synthetic RNA into the liver of a non-human primate to see if it would, sort of initiate the complete process. And basically, after a number of failed attempts, there were two things that were actually key for getting this to work. One of them was we actually found a missing piece at the end of the genome of the virus that turned out to be very highly conserved and absolutely necessary for, a functional sort of genome to replicate. And then the other thing was actually dealing with the incredible heterogeneity that hepatitis C has. We estimate that in a single chronically infected individual, there's about 10 to the 12th variants produced per day.
Wow.
There were an estimate of about 200 million people chronically infected with this virus around the world. So that's a lot of diversity. And so and as you probably know, these RNA viruses, including coronaviruses, make mistakes. And part of the way that they survive in nature is to create these populations of variants. But some of those variations can be lethal, sort of for the virus. So the other thing that was really the key to success was to sequence multiple sort of cDNA clones for the virus and actually assemble a sequence which was basically the preferred nucleotide at every position, every one of the 9,600 positions in the viral genome. And when we made that clone and tested it in animals, it worked. So we knew that we finally, had a functional hepatitis C virus genome, and that was really, it didn't exactly translate into being able to replicate the virus in the lab. That came a few years later, but it was based on at least sort of starting with what we knew was a functional clone, and then coming up with some variations that allowed it to replicate in cells.
Well, that's well said in terms of a layman. I know it's probably hard to summarize that complex science, but I think that's very understandable for those of us who aren't at that level. And the next kind of big accomplishment of yours was discovering how the virus infects the liver cells and then producing agents which can block it, thereby, forming into a chronic disease. Tell us, what is the key step towards the virus infecting liver cells, and how did you target that with blockers, with therapies?
Well, I think that there are a couple things that are maybe separable here. Sort of one of the things that, we were very interested in, sort of understanding how the virus got into cells and why it prefers to replicate only in liver cells, hepatocytes, the sort of main cell in the liver. And you could imagine that, sort of one way to, block entry of the virus would be to use antibodies that could actually neutralize the infectivity of the virus, or antibodies that would bind to the receptor. But actually, the key to success for developing drugs for hepatitis C wasn't really the entry process. It was actually this intracellular amplification machine that the virus used to make more copies of its genome RNA. And there were, basically three components of that were very important for that machine to function. One of them was a viral protease, something which, sort of cleaves proteins. And that was absolutely required for assembling a functional replication machine. And then the other one was a polymerase, the target of drugs against HIV and HBV, a polymerase that makes RNA. That was another, sort of favorite target. And then there was another protein involved in replication called NS5A. And those really turned out to be, sort of the poster children, if you will, of the eventual sort of antiviral drugs that we use today. And the ability to actually develop those drugs, I didn't do it. That was done by, people in biotech and pharma, but was a cell-based system called the replicon system. And basically this was a system where for the first time, and this was done by Ralph Bartenschlager and Volker Lohmann, they actually sort of isolated a human liver cancer cell which had continuous replication of HCV RNA in that cell. And that was the first time that people had been developing compounds that inhibited the polymerase or inhibited the protease, but there was no way to find out if these would actually get into a cell and inhibit the ability of the virus to replicate. And so that replicon system, which was, developed, more than 10 years after the virus was discovered, really became kind of the engine of drug discovery.
Wow. I mean, it's such an amazing journey. And I think you guys have gotten to the point now where it's a controlled chronic disease as opposed to a lethal condition. But I guess the next question is, will there be a vaccine such that people can prevent from ever being infected from hepatitis C?
Well, I would say turn it around a little bit in the sense that the drug cocktails that we have today actually can eliminate the virus from an infected individual. The virus can actually be eradicated, and that happens in probably about 98% of the patients that are treated. And it's basically 2 or 3 months of an oral therapy, and the virus is gone. And even
no further treatment needed. Just 3 months of treatment. It's eradicated. It's not lifelong therapy.
No. So it's not like HIV. It's not like hepatitis B, where if you take away the therapy, the virus comes back. That is usually not the case with hepatitis C, and we do believe that the virus is gone in the vast majority of people that are treated because, often, people will contract cancer and they could be on, very sort of tough anti-cancer regimens that compromise their immune system. So even people that become, for instance, severely immunocompromised, we don't see a resurgence of the virus if they've been successfully treated. That's and wonderful. It's even allowed, people to use organs from Hep C positive people for transplantation and, sort of treat the transplant recipients and eradicate the virus. Now, that doesn't mean that the disease is done, because if you've been chronically infected with hepatitis C for 40 years or so before you you get treated, your liver may not be in tip-top shape. And so we are seeing that, patients that are cured of the virus can have an improvement in liver function. Fibrosis does seem to be reversible. But people that have progressed to more severe liver disease, this sort of nodular sort of scarring called cirrhosis, really are still at increased risk of developing liver cancer, which is really, sort of the problem with chronic hepatitis C. And so they still have to be monitored fairly closely.
Okay. But I mean, it's still great to hear. I didn't know that it could be eradicated the way you were saying. That's absolutely phenomenal.
Yeah, it's.
But switching gears back to COVID, which I think kind of has monopolized everyone's thoughts the last year to year and a half. As a virologist, obviously COVID kind of hits close to home to you. What was your perspective on when the pandemic started, and how did it affect your lab work?
Well, and it still is. I think we certainly recognized in January of 2020, shortly after the first of the year, that sort of something was going on that we should pay attention to, and that just sort of grew over time. And in mid-March, we were facing sort of increased hospitalizations and cases here in New York. Rockefeller University essentially shut down, and only the laboratories that were actually working directly on COVID-19, SARS-CoV-2, coronavirus research were allowed to continue with, basically a trimmed down sort of skeletal crew. And it really has been amazing to watch, sort of how this has unfolded around the world. But even in our sort of microcosm here at Rockefeller, we had already been working on coronaviruses before this new virus showed up. And so several people in the lab, really stepped up to the plate, and we began to, learn how to work with SARS-CoV-2 and sort of other coronaviruses. We have a containment facility here. We began to do screens to look for, sort of cellular factors that were important for the virus to replicate. Really initiated a large number of projects that involved, some of your alma mater folks over at Sloan Kettering and at Cornell and at NYU, and formed kind of a COVID-19 working group. And so rather than sitting at home twiddling our thumbs, and, we really, there were a core group really working very, very hard on this virus, and still is. And so during that time, the work that we were doing on hepatitis C or on hepatitis B or on other aspects of virus-host interactions basically ended up sitting in the freezer. We couldn't do any of that work, and actually some of the experiments that were in progress couldn't even be continued. So we're really right now just in the stage of restarting, some of the work that had to be shelved. And I guess we've got a bit of a train wreck in the sense that we still have all these coronavirus projects going on at the same time. So it's been a busy time for virologists. I would say that, yes.
Yeah, for sure. I mean, you guys are kind of front and center in the media every single day. And, really along those lines, as someone who has worked with viruses his entire career, you focused on vaccines, you know how difficult it is to study viruses, form vaccines. From your perspective, how amazing is it that a vaccine was formed in less than a year for COVID-19?
Oh, I think it is amazing. And, one of the great benefits of this, I think there is a silver lining here, is the exploration of new technologies for vaccine development, like the messenger RNA vaccines that have been approved for emergency use here in the States and elsewhere. And so I think and we're going to get more information on how people respond to these vaccines from the COVID-19 vaccine effort that I think, and I hope, will sort of get us over the hump for hepatitis C, because it's now, I guess, a little bit embarrassing that, for COVID-19, vaccines were available in less than a year, and people have been working on hepatitis C vaccines for, sort of approaching 30 years. But I think some of the technologies that are being applied to COVID-19 look very attractive for hepatitis C. Of course, hepatitis C is a different kettle of fish when it comes to viruses. I mean, it is a virus that can establish a chronic infection where, coronaviruses are typically acute resolving infections. And the natural history of hep C infection is such that even people that resolve the infection naturally, and that's about 30% of the people that are infected with the virus, are still able to be reinfected. So there's not protective immunity. Whereas I think if you're a convalescent, sort of COVID-19 individual, your chances of getting infected with, clinical symptoms or hospitalization is very low.
Yeah. I mean, it's great to hear your perspective, because we all understand theoretically how hard it is to make a vaccine, but most of us are not in there every single day trying to form one. So it's great to hear from you, because it really is stunning to see how quickly the world mobilized, collaborated, worked together, and then formed this vaccine in such a record fashion. But wrapping up here, I just wanted to, obviously your work is so inspirational. What would you say to young scientists that are perhaps in college, in graduate school, doing their postdoc, who want to follow your footsteps? What's your advice for future scientists?
Well, I think it's important to sort of find a path that you're really passionate about, that this is what you want to do no matter what, and pursue that. Because I think that's where you sort of probe the depths of your curiosity, and you also have the commitment to overcome obstacles that invariably pop up. And I think the hepatitis C story is, an example of that. Something where the sort of mystery virus, it took 15 years to find it, and then it took another, sort of almost 30 years or 20 plus years to develop therapies. That's actually fast, but it did involve a series of breakthroughs that basically presented another obstacle that had to be overcome. And so you've got to be a little bit pig-headed and persistent. And I think enjoy the journey. I think as biomedical scientists, we're really fortunate to be able to do this, what we call work, but it's actually our hobby. This is what we want to do, and this is what we would be doing, if we weren't getting paid to do it. So the fact that you can actually have a job that sort of pays you a salary, but it's actually sort of your hobby. That's pretty special. So I think if you can shoot for that, you'll be in good shape.
I could not echo those sentiments more. I always tell people I haven't worked a day in my life. Come to work and you love what you do. It makes things much easier. And drive, focus, passion, those are all things that kind of lead to the kind of success that you've had. And so, wrapping up here, I know it's been half an hour and you're obviously very busy. Charlie, thank you for joining us. Your work is truly inspirational. I'm sure you get told this all the time, but your work has basically saved millions of lives, millions. And it's really changed the world. So kudos to your team, kudos to you, kudos to all your students and everyone who you kind of motivate and you mentor. And it's a real pleasure to speak to you.
Well, thanks very much. I really enjoyed it. And I think that you're also an incredible role model. I sort of looked up all the stuff that you do, and it's unbelievable. I guess you must be one of those people that only requires 15 minutes of sleep a night or something.
I wish that was the case. Unfortunately not, but but I wish that was the case, and I appreciate that compliment. And again, thanks for all you do. Truly inspirational, Charlie. Thanks for your time. Have a great afternoon.
Okay, you too.